So it was my task to kind of give you a talk on, what is or define the disease memory in psoriasis. So these are my conflicts of interest. So just to kind of, I know you're all familiar with this kind of a scenario. You have patients with like moderate to severe psoriasis, and you put them on a treatment, and they do really well. Their skin clears, and sometimes almost completely, if not completely. But always when you take the patient back off the treatment, the psoriasis comes right back. And what's striking about that is that the disease almost always comes back up in the same areas that were affected prior to treatment. And the question is, is this state here where you get a complete clearance, is the skin completely normal? And this was one of the reasons why Jim Kruger, this is a publication from him about 11 years ago, that kind of was the first one to define the molecular scar of psoriasis. And this was really the first sign that there might be some kind of like an inflammatory memory in psoriasis. So this was a microarray based study on patients with moderate to severe psoriasis treated with etanercept. And what they did is they did a baseline biopsy, and then they did a follow-up biopsy on completely resolved skin about 12 weeks later. And you can see in this table over here, this is the baseline change. These are the differences or normalization at week 12. But what really stood out was that not all the genes actually improved. There was a significant proportion of genes that had less than 75% improvement, even though the disease was clinically gone. And many of these disease-related genes had important pro-inflammatory functions, such as interferon signaling, IL-22, IL-17, and interferon gamma. So if you look at this, the magnitude of this molecular scar, you can see the big circle is what the transcriptomic changes looked like at the baseline. And then you have two treatments, two biologics, one anti-TNF, etanercept, the other one was the Kinemab, the anti-IL-12, IL-23 agent. But what you can see is that the residual signal that is still in the non-lesional skin accounts for about 20% of the overall transcriptomic changes. And if you look at what are the biological pathways that are still active in that plaque, even though it is still is subclinical, a lot of that has to do with inflammatory processes that are highly relevant to psoriasis pathogenesis, such as the role of IL-17A in psoriasis, macrophages and fibroblasts, and some neutrophil-related processes. So what is the biological mechanism behind this molecular scar? So I would consider this to be a landmark study. It's actually now 18 years old. This is a study that Frank Nestle did. And he was utilizing a xenograft model. This is where you take skin from healthy individuals or completely resolved psoriatic skin and also healthy control skin. And they grafted that onto these immunodeficient mice. And you can see the skin, what it looks like at baseline. This is kind of healthy, normal looking skin that you would see any place. And here you have it grafted onto the mouse. But what happened within about 4 weeks is that the non-lesional skin in the psoriasis individuals developed what looked like a psoriatic plaque. They had marked epidermal hyperplasia, whereas the control skin did not have any changes at all. And this tells you that there's some element in the resolved or non-lesional psoriatic skin that is kind of poised to get activated and actually turn the skin back into psoriatic plaque. And you can also see that here, this is a papillomatosis, which is kind of the degree of epithelial proliferation. You can see in the psoriasis, non-lesional psoriasis grafted onto the mice, you get marked activation and epidermal thickening, whereas in the controls, you really do not see any changes at all. So these results kind of suggest that there are resident immune cells in non-lesional or resolved psoriatic skin that when activated will initiate psoriasis development. So next slide. So this is a study that was done like a few years later by another group led by Liv Eidsmo in Stockholm. And what they did is they looked at patient skin, patients that had been on biological treatment, on UVB, and then compared that to healthy normal skin or active psoriatic skin. And what they found is that there's increased number of CD8 positive T cells in treated skin, not as much as in active psoriatic skin, but increased. And if they focused on the CD49 positive population, they could see that in biologically treated, non-lesional, completely resolved skin, there was increased number of the CD49 positive CD8 T cells in the epidermis. So next slide. So what are those CD49 positive cells? So CD49 is the alpha-1 subunit of the alpha-beta-1 integrin. This is a molecule that is critical for trafficking of immune cells into the skin. And in this study, they used the same model, the xenograft model, where they grafted the non-lesional resolved psoriatic skin onto these mice, and then they treated them with a drug to kind of inhibit the alpha-1 integrin. In the isotype treated group, they got a full development of psoriatic-like lesions, whereas actually if they blocked alpha-1 integrin, they could completely prevent the transition into psoriasis plaque. It's shown over here also. This is the acanthosis, which is the measure of the epidermal thickness in psoriasis. Pretty marked prevention of development of psoriasis. Epithelial genes that are reflective of epidermal activation in psoriasis, such as keratin 16 or IL-8, which is CXCL8, neutrophil chemokine, completely prevented. And then also the papillomatosis index was completely normal. And if you looked at what happened in the skin, and this is kind of looking at the epidermal T cells, you can see that by using this treatment, they could completely prevent the accumulation of T cells into the epidermis. So this means or indicates that epidermal TRMs, or tissue-resident memory cells, are important in psoriasis development, and they really need to get into the epidermis to really mediate their effects. So they need to be in the epidermis. So next slide. So what are those T cells doing? What are they poised to make? So this is kind of taking the cells from resolved psoriatic skin. This is from individuals on biological treatment. Some of them had been on biological treatment for years, if not decades. They took out the T cells and then they activated them. And when they did that, you can see that almost all of them, even in the resolved psoriatic skin, were poised to make IL-17A. Not quite as high as you can see in active or UVB treated skin, but quite significantly and obviously much higher than what you would see in healthy skin. They also make a little bit of IL-22, but that was much less significant. And it was only the epidermal CD8 positive T cells that had this predisposition to make IL-17 on activation, not so much the dermal CD8. So this is consistent with that the epidermal CD8 positive TRMs are the ones that are really poised to get activated, make or secrete IL-17, and then promote a disease recurrence. So please, next slide. And if you look at kind of a deeper phenotyping of these cells express the cutaneous leukocyte antigen, the CLA, so that indicates that they're able to home into the skin. They also express the TRM marker CD103, which is alpha E beta 7 integrin that actually allows them to adhere to the epithelium and stay put. And interestingly enough, they express very high levels of both CCR6, which is the chemokine receptor for CCL20, which is a chemo attractor for Th17 or TC17 cells. And they also have very high expression of the interleukin 23 receptor, indicating that they're very responsive and may even require IL-23 for their function. Next slide. So what's the model for disease memory in psoriasis? And this is focused on the tissue-resident memory cells. So in healthy skin, you have some T cells. It's normal to have some T cells just normally at baseline in skin. But when you develop psoriasis, you get marked increase in IL-22 and IL-17 positive TRMs. And these home into the epidermis. Whether it's with treatment or spontaneous remission or or successful treatment, you have resolved psoriasis, that clinical disease goes away, but these cells kind of stick around. They're inactive, but they're lodged into the epidermis. And if you have the disease trigger or you remove treatment, these cells are primed to kind of get activated again, make local IL-22 and IL-17, and then recruit additional circulating T cells into the tissue, and then recreate and reestablish the active disease all over again. So go to the next slide. This is still not the only mechanism that we think is playing a role in disease recurrence in psoriasis. There is some evidence for epigenetic changes. This is much more or much better established in mouse models of inflammation. So this is a review article that was recently published in Cell. This is from the Elaine Fuchs group, who's been kind of leading the field in looking at this. And what they found is that in keratin epithelial stem cells, they can actually find these epigenetic modifications. So in normal skin, you have the chromatin kind of being tightly packed, inactive. When you have an inflammatory response, you actually open up the chromatin, you get transcriptionally active, that kind of then mediates production of pro-inflammatory genes. When you treat the disease, or whether the initial inflammatory process goes away, there's a resolution. These changes on the chromatin level do not go away. They do not disappear. So you have them there ready to go. And it's only if you have a recall or a reinitiation of the disease process, the chromatin, the DNA changes are there ready to go, and you get a much more rapid and augmented transcription of these genes, and then ready to kind of recreate the disease process. So these play a major role likely in disease recurrence. It's still very poorly understood in psoriasis, and actually has not really been studied in resolved psoriatic skin. So go to the next slide. So just to kind of quickly summarize, so the disease memory in psoriasis, I'm going to see if my remote actually worked. So the CD positive tissue-resident memory cells play a key role in the disease memory in psoriasis. They express CD103 and CD49A, and that helps them to anchor them in the epidermis. They're poised to produce IL-17A on activation. And they express high levels of the IL-23 receptor, suggesting that IL-23 has a role in the maintenance of TRMs in psoriasis. CD8 positive TRMs persist in skin for a very long time, and we're looking at years to decades, and they even persist despite successful treatment. Epigenetic changes, like I showed you on my last slide, chromatin remodeling has been described in the epidermis. The data still is mostly from mouse models, but they have been shown in that particular setting to amplify inflammatory responses on recurrence. And both the TRMs and epigenetic modifications in stromal cell types are likely both to contribute to the molecular scar of psoriasis and to disease relapse on discontinuation of treatment. And I would say that both of these mechanisms provide a target for a cure, or at least a reset for psoriasis. So I think it's going to be very exciting to see how the field goes in the future.